new technologies to study cell differentiation. So this webinar is recorded, and we will share with you a link afterwards if you want to take a look again later on. And today we are having a presentation first, and then a question and answers session afterwards. You can already type your question during the webinar, or in the end you can raise your hand and I will give you access to the microphone so you can ask your question directly. My name is Katja. I'm the application scientist at 4Dcell. Just a little bit of background about myself. So I did my PhD in biomedical sciences at Karolinska Institutet in Sweden, and my main expertise is in the area of immunology. Today I'm going to be one of the presenters together with Patricia Davidson, my colleague, who is the R&D project manager. So Patricia has a PhD in chemistry and biomaterials at CNRS from France, and her main expertise is in microfluidics. Joining us for the question and answer session is Rita Ribeiro, our Chief Technical Officer. She has a PhD in physics by the University of Porto in Portugal, and her expertise is in microfabrication and optics. So before anything, just a little background on 4Dcell. So it's a biotech company located in Paris, in France, and we develop in vitro assays for the control of cell microenvironment. So the idea is to provide bioengineered tools that researchers can use to acquire more relevant and accurate readouts on their cell culture. And to your right, you can see an image of our facilities, specifically of the clean room that we use or where we produce our tools and devices to send to our clients. And a picture of Patricia working on microchannels and all dressed up as it should be on clean room. And so moving on cell differentiation, just to give you also an overview, cell differentiation is a process which involves a sequence of events that starts by from an embryonic precursor, which leads to the formation of germ layer intermediate cells that later on are differentiated in lineage restrictor progenitors, which ultimately are differentiated in terminal cell types. And one example I have here are the mesenchymal stem cells that can be differentiated in, for example, the mesodermal lineage, and thereby becoming, for example, adipocytes or chondrocytes, osteocytes. And another example as well that we are going to talk about later on are the hematopoietic stem cells. And those, as you know for sure, is those cells, they lead to the lineage of to the lymphoid lineage, for example, B and T cells, and the granulocyte lineage, which, for example, leads to the formation of monocytes. And I'm giving the example of monocytes because cell differentiation, you can study at different stages, right? And for example, monocytes, depending on the cell culture conditions that you use, can be differentiated in osteoclasts, dendritic cells, and macrophages. And of course, cell differentiation is a process very important to study, and especially nowadays that there is so much going on regarding cell-based therapies, and also, for example, for wound, for healing medicine or regenerative medicine. Thus, what we would like to give you a presentation about today will be how to do certain studies using 4Dcell products that can be, for example, focused on the research of reprogramming stem cells, studying pluripotent stem cells, but also progenitor cells, and as well as cardiomyocytes and muscle cells. But to start with, I will give you an overview of the different technologies that you can use to this end. And we can start by the micropatterns, which are substrates that can be chemically modified, where you have cells attaching to specific areas, and there are other areas surrounding these patterns, which are non-adhesive. So the idea is that the cells, once seeded in the micropattern areas, they will adopt the shape of that particular pattern. And there are different shapes that you can use. For example, lines, squares, triangles. Here we have the example of lines, and also on the bottom you can see a cross-sectional image of a micropattern with a cell on it. So these are our standard micropatterns, and the cells in these areas can either attach to bare glass, but also you can use extracellular matrix to promote the cell adherence, like fibronectin or other. So this, in addition to these standard micropatterns, we also have the dynamic micropatterns, which are more based on the click chemistry. That you can use, for example, for co-cultures. And Patricia is going to talk a little bit about this later on and give you an example. But the basic idea is that you have adherent areas where you seed your cells, and at some point you can add a protein which will render the non-adherent areas adhesive, and then you can see the second cell line, and from there perform your studies. So here you have an example of C2C12 cells in line micropatterns. We will come back to this as well later on. So the micropatterns are typically provided in glass cover slips or glass bottom Petri dishes, but as well in a multi-well plate format, 384 or 96. But many people, they would like to produce the micropatterns themselves in the lab, and for that we have a fabrication kit that allows you to do everything yourself in a simple way in your lab. So the next technology that I would like to introduce you to are basically substrates which contain topographic features, so-called micropillars, and these features or these topographic features, as I was saying, they are made in PDMS, which is an elastomer. So the cells are seated on top of these pillars, and the pillars can have different diameters. So the larger pillars, they are stiffer compared to the thinner pillars, and the cells can sense that, and they will behave differently depending on the stiffness of the pillar they encounter. And this is very important in cell differentiation, because it has been shown that depending how stiff the pillars are, the cells may move to a different stage of differentiation, or they can even try to invade the space between the pillars. Here you have a scan electron microscopy image of a cell sitting on top of two types of pillars. You can see the different diameters, and of course this is rather an impressive image. It's not what you get from an optical microscope, but it illustrates the idea that I was explaining earlier. And the micropillars are usually provided in small glass cover slips. So moving forward to the next product that we have, or device, which is based on structured surfaces. Basically what you have are glass cover slips, sorry, which are coated with a soft or a hard material, for example, a soft gel that can be polyacrylamide, or an elastomer like PDMS, as I mentioned before. And these substrates, they can be microstructure or not, just flat without any shape. But you can choose to have a specific shape. In this case, we have grooves, but it's possible to customize designs, or to have, for example, wells or squares, if you want your cells to be seated on these structures. And here you can see better how it looks like. So you have this material, the gel or the PDMS, and the cells are some of the cell types, they choose to be sitting inside the grooves, but actually some other cells, or at different stages of differentiation, the cells may prefer to be lying down in the middle of the or in between grooves, for example. So these structures, they are usually provided also in regular glass cover slips. So moving to the fourth device I would like to introduce you to. This is what we call a cell confiner. And basically the idea is that with this device is that cells are compressed between two parallel surfaces. So how does this work? You can see here an image of, for example, you have your cells on a petri dish or plate. And on top, you are going to apply an assembly or a system which is composed by a cover slip, a glass cover slip with the micro pillars of a specific height, which is attached to a transparent piston, also made of PDMS. So when you apply this system on top of the cells, they will compress to the height that you predefined. So you can choose how high these micropillars are going to be, and therefore that will be translated on how much the cells are compressed. Usually researchers use pillars of between 1 to 20 micron. But for example, if you're interested in confining bigger organisms or embryonic explants, for example, it's also possible to have pillars with a higher size or a bigger size. For example, here you have HeLa cells in conditions where they are not confined. And once they are confined to a height of 5 micron or 3 micron, and you can see the difference among them. As you can see, when they are more compressed, there is formation of blebs from the cellular membrane. So the cell confiner can have different applications for cell differentiation. Also, Patricia is going to give you some examples and possibilities that we can try to adapt to this field. And we provide the cell confiner in two formats, either the static, 1-well or 6-well, which are compatible with the petri dishes or 6-well plates. And we have another system, which is what we call the dynamic confiner. And in this case, the system itself is connected to a vacuum pump. So the static confiner, it's the user who regulates or basically handles the system itself. So you have the cells on the plate or the petri dish, and then you have a modified lid with the piston I was telling you about, which is transparent, and the cover slip with the pillars of a certain height, and then you can close the system yourself. In the case of the dynamic confiner, it's a little bit more complex because, as I said, it's connected to a vacuum pump. But the advantage of this system is that allows you to see the cells being confined in real time. So basically the moment that you are confining the cells, whereas these two systems only allow you to see the cells already confined. In the case of the dynamic confiner, the principle is the same as for the static. You have your cells on a petri dish, and then you have a suction cup, which works as the piston attached to a cover slip with a specific micropillars with a certain height. And then you just need to close the system and compress the cells. So now that I presented you the different application, actually the different, I'm already stealing Patricia's part. So that I presented you the different products we have, I'm going to pass the word to Patricia, who is going to give you some ideas with applications that you can work on in this cell differentiation area.
Okay, thanks, Katja. So yeah, I'm going to be walking through some of the applications that we have for cell differentiation. The first one I'm going to show you is actually the opposite of cell differentiation. It's reprogramming cells to become more pluripotent. So here I'm showing a result from one of our clients. So we're always very proud to show results that our clients have obtained. And in this study, they studied how prone cells are to reprogram. So essentially, if you want to reprogram somatic cells to pluripotent stem cells, there are you can reprogram them with more or less efficiency. And these researchers found that the MLK1 activity is important for reprogramming cells. And as you can see here, they tested different shapes and sizes to see if this had an effect. And in fact, they found that the MLK1 activity was the best in the smallest micropatterns, as you can see here. And so in this case, the MLK1 was more cytosolic than nucleoplasmic. So there is an important effect of the cell size also on the reprogramming efficiency. In this case, these researchers looked at, again, micropatterns and their effect on reprogramming. And so they seeded cells in the micropatterns, and after 10 days, the cells were forming spheroids on these micropatterns, and they saw an increase in the amount of alkaline phosphatase that the cells were expressing. And so they looked at RNA-seq to understand what was happening with the cells, and they found that these cells were expressing more and more of the genes that are typical of embryonic stem cells or induced pluripotent stem cells. So it really appears that these cells, simply by laterally reducing the amount of space they can take, are in fact reprogramming themselves to become more stem cell-like. And so they showed this with a schematic that I'm showing here. So really just with the micropatterns and forming, laterally confining the cells, they were able to give them more of a stem cell type phenotype. So now we're going to talk about differentiation. So first we're going to start with pluripotent stem cells. So in this study, the authors used relatively large micropatterns. So they're 1,000 micron large, and you can see here are many, many cells on this micropattern. And they seeded human embryonic stem cells. And so you can see here they used 3 different markers for pluripotency. So the cells were all pluripotent at the beginning, but they added BMP4 and waited 48 hours. And they found that in this case, the cells imitate the gastrulating embryo organization. So what you have at the periphery of the micropattern, the cells turn into triploblasts. And then as you go towards the interior of the micropattern, you have the mesoderm and ectoderm lineage. So it's really imitating the different lineages that you would find in the organization of a gastrulating embryo. Here I'm showing an example where researchers took a dissected midgut from Drosophila, so a fly, and they looked at intestinal stem cells. And so what they did is they wanted to understand whether pressure in the gut could have an effect on differentiation. So they used the microfluidic device and compressed the midgut, and they found that the intestinal stem cells were being activated. And this was turning on quite quickly. As you can see here, within a matter of 20 seconds, you were able to activate or deactivate the cells simply by adding force. And so with this system, they were able to show that the intestinal stem cells have a preferred differentiation towards the enteroendocrine cell lineage under mechanical stress. So now we're going to talk about cells that are a little bit more differentiated towards a specific lineage, so progenitor cells. So the first type are mesenchymal stem cells, which are probably the most commonly studied stem cells that you've probably heard about. There are many, many studies showing that the stiffness of the surface will impact the differentiation of the mesenchymal stem cells. And so here I show some examples. These are all covered in the review that I'm showing at the bottom, but essentially the stiffness of the surface will allow the cells to differentiate more easily towards a lineage that is similar to the stiffness that you find in vivo. So for example, neuronal or adipogenic lineages will be preferred on softer surfaces, whereas bone cells will prefer to differentiate on stiffer surfaces. You can also recapitulate this using micropillars. So as I'm showing here on the top, short pillars will feel more stiff to cells, whereas really tall one will feel deformable and soft to cells. And so if you put mesenchymal stem cells again on these types of pillars, you will see that they will preferentially differentiate to adipogenic cells, so fat cells, on the taller pillars, but they will preferentially differentiate to bone cells on the shorter pillars because they sense the stiffness. So now we can talk about hematopoietic cell differentiation. So essentially blood cells that are made in the bone marrow. So you can see there are many, many different cell types here. And one thing that's important to note also is that there are many different types of stiffness in the bone. So bone is probably the stiffest part of your body. At the bone surface, you have a stiffness of about 40 kilopascals. And then you have the perivascular niche, which is basically the vessels that go through the bone, which are softer, around 3 kilopascals. And then you have the marrow itself that is very, very soft, around 0.3 kilopascals. So you really have a very wide range of stiffnesses in the bone. And we're only now starting to understand how these stiffnesses can direct how blood cells can differentiate. So here, these researchers studied the phenotype of hematopoietic stem cells, and they found that on softer surfaces, the stem cells stay very round, but when you start putting them on stiffer surfaces, they start forming protrusions. And in fact, they showed that if you put the cells on surfaces of different stiffnesses that are similar to the perivascular niche or bone, you will have commitment to different types of lineages, as we're showing here. Here's another example of progenitor cells. So in this case, cells from your teeth. And so these researchers wanted to understand whether compression, as you might have in your teeth, can affect differentiation of cells. And so they used human dental pulp stem cells and compressed them. And they found that there was a preferred amount of time and a preferred amount of pressure that the cells needed in order to differentiate to odontoblast-like cells. And so here it's quite an interesting experiment because they were also able, at the same time as they were compressing, to use a 3D topography that was similar to the in vivo topography. So they were really imitating the in vivo conditions both ways. So now we can talk about cell lines that are committed to a specific lineage. So first we'll talk about cardiomyocytes. So here I'm showing some results that were obtained by a collaborator of ours. And so we see here, once cardiomyocytes are seated on flat surfaces, you have a random organization of the cytoskeleton. But if you force them to adhere to line surfaces, they will organize their cytoskeleton, and you can see that the sarcomeres are very nicely aligned in this case. You can also put them on rectangles, and so they will have different aspect ratios. And in this case, the researchers found that the best conditions were to put them on a rectangle that had an aspect ratio of 1:7 on surfaces that were 10 kilopascals stiff. And in this case, in the most elongated cells, they also were able to exert the strongest forces. So it really shows that the sarcomeres were well aligned, and you had the most effective force transmission in this case. Another important point about cardiac cells or cardiomyocytes is that in vivo they aren't naturally present by themselves. They have a lot of other cell types around them that secrete factors and that probably help them differentiate going from the fetal heart to an adult heart. So you can see here you have cardiac fibroblasts around the cardiomyocytes, conducting cells, endothelial cells. So we think it would be a great project to use these micropatterns, but specifically the dynamic micropatterns. So in this case, you would first see the cardiomyocytes and have them at the right shape. And then you could activate the rest of the surface and add cardiac fibroblasts or endothelial cells so that they can secrete the factors that will help the cardiomyocytes differentiate. So next we'll talk about muscle cells. So here's a beautiful picture obtained by our collaborator Bruno Cadot. In this case, he differentiated C2C12 mouse myoblasts to form myotubes. So C2C12s are a very common cell line, but they don't differentiate fully to myofibers. So here you can see very nice myotubes, but in order to differentiate to myofibers, you can use primary myoblasts. And in this case, you can see the different stages. So the myoblasts will fuse to a myotube. Following this, the myotube, the nuclei will spread in the myotube so that they're evenly spaced. And then they will have a stage where they go from the interior of the myotube to the exterior. And so this is something that we can see in vitro in experiments using soft surfaces. So here's a video that was taken where the primary myoblasts were plated on a soft surface. And so you can already see the nuclei spreading, going back and forth. And then as the cell differentiates more and more, you can see that the nuclei are popping out of the myotube very nicely here. So again, the important points here were that they used primary myoblasts, they used agrin, but they also used a soft substrate that helped the myotubes differentiate. So as a take-home message, I've shown a lot of applications, whereas you can use stiffness, the topology of the surface, you can confine cells on micropatterns, you can exert forces, and all of these can have a very significant effect on how well your cells are able to differentiate. And so I hope that you found this webinar useful. Our next webinar will be on wound healing. And if you have any questions, I will be happy to answer them.